Dr. O.M. Germany. Thank you. Okay, so Dr. O.M. in Germany, he will be speaking to us today about on understanding social interaction disorder. What can worms teach us? So once again, I'm going to call on Dr. Germany to please come here while I read the citation. Thank you, sir. So, Dr. Omamweobi Mishak in Germany, or simply called Mamus in Germany, is a neuroscientist and anatomist, an academician and a bio-research strategist with a strong passion for advancing brain health in Nigeria and globally. He is an assistant professor of anatomy and neuroscience at the University of Medical Sciences, Ondo, Nigeria, where he leads the Laboratory of Experimental and Translational Neurobiology. He is also the Deputy Director of the Central Office for Research and Development, CORD, at UNIMED. Further, he is a visiting assistant professor at the Albert Einstein College of Medicine in New York, USA. He was previously a senior faculty and researcher here at FUTA at the School of Basic Medical Sciences, where he provided research mentoring and support to many young faculty members who have ultimately led the establishment of the FUTA Neuroscience Group. Dr. Germany obtained a BSc Human Anatomy in 2006 at the University of Port Harcourt and an MSc and PhD in 2011 and 2015, respectively, at the Obafemi Awolowo University, Ile-Ife. He held a postdoctoral position from 2017 to 2018 at Albert Einstein College of Medicine in New York, USA, and his research activities are focused on understanding how the brain is affected by an interplay of genetic and environmental factors that could cause perturbations and or death to the brain cells at critical stages of development and the lifespan. Dr. Germany has received several fellowships and grants, including the International Brain for Research / International Society for Neurochemistry Research Fellowship, the Ebro Return Home Fellowship, the ISN CAN 1B grant, the Young Ebro Award, the Alexandra von Humboldt George Foster Experienced Researcher Fellowship, as well as the Ebro Early Career Award and ISN Career Development Grant, amongst others. He was named the Professor James Kamali Lecture Awardee in 2023 by the Society of Neuroscientists of Africa, which is the foremost neuroscience society award on the continent of Africa, and received the Colin Blackmore Bursary Award from the Royal Society UK in the same year. Presently, he holds the Emerging Global Leader Award from the National Institute of Health, NIH USA, which is a 5-year grant leveraging the innovative C. elegans model to study gene-environment interaction in autism spectrum disorder. I think we don't know about this research, but at least this research is worth $500,000 US. Dr. Germany holds several academic leadership roles and serves on numerous scientific committees, both nationally and internationally. He is a member of the ISM Career Development Committee and has served on the NIH Neurotoxicology and Alcohol Studies section. He has authored over 90 peer-reviewed articles and book chapters in high-impact journals and book series. He is actively involved in the mentorship of undergraduate and postgraduate students, as well as early career scientists, with many of his students and research mentees going on to win numerous grants and fellowships. I'm proud to say that I am one of those mentees. Welcome, Dr. Ijomoni.
Good afternoon, everyone. So, I will be opening the session to Dr. Ijomoni's plenary lecture. Thank you.
Good afternoon, everybody. Good afternoon, Prof. I was telling them that I don't know how they put my lecture after that powerful lecture in the morning. If you just go home for today, and you don't allow, maybe we should talk first before the fathers talk. It's easier. So we don't have to try to overdo too much. Okay, Prof. I'm really glad to be in the presence, and I'm glad to be here with my lecture. I appreciate it. Our first time, no, that was the first time, that was the first time we had an interaction, and I appreciated your support. And fun is more, the idea is good tree. It's something we are developing already. I'll probably mention it towards the end of the lecture. I learn a lot. So sometimes when somebody tells me something, I go back to the lab and try to improve on it. If you stay to the end, I'm interested in it. So today, much ado about nothing, I'm going to give this talk. I had to modify the topic, you see, to social phenotypes in developing brain disorders, what can one teach us. Gratitude to my dean, provost, and the chair for inviting me back here. It's nice to be here. It shows that at least when I was here, I was good. Because if I was not good, they would not think of me to call me out here. So, first of all, some definitions. Let me just quickly do some basic definitions. What is a phenotype? That is an individual's observable traits, or what we know ourselves to be, are determined by our genetic makeup and by the environment. How tall you are, how short you are, your eye color, these are phenotypes. So what are social phenotypes? These observable traits in the context of social behavior, how we interact with people and our environment. And they are shaped as well by genetic predispositions and environmental factors. So what is developing brain disorders? I didn't want to use the big name. Big name is just simply neurodevelopmental disorders, which is basically diverse conditions that result from anomalies during brain development. And basically that's what most of my research currently tends to do. So while I'm talking about this today, most of the developing brain disorders have a social context, or what we could say a mental context. Brain development, when impacted at early stage, impacts a person's well-being as well as how the person relates to within his environment. So we have social behavior that broadly defined as interaction between an individual of the same species and social interaction deficits, which are basically disorders or dysfunctions in this interaction that we expect. For example, I'm sure while we've been here, Prof has a very good social skills. So yeah, maybe that's why it's pretty young. He was telling me he's still pretty young. After the whole meeting, we see all of us, we're interacting one another, you're checking, even if you are introvert, that's not the disorder or extrovert, but you're also able to communicate within the group of space you find ourselves. So that's social behavior. And deficits will come from this behavior that encompass a wide varieties of neurodevelopmental disorders and and psychiatric conditions. And a characteristic hallmarks of these are difficulties in trying to communicate, difficulties in interacting, difficulty in responding or giving feedback to interactions. So examples of these include the autism spectrum disorder, attention deficit disorders, schizophrenia, intellectual disability. I think here I'll take a pause. Those days we used to call them, okay, Prof is the historian, so I'll try not to do history, I'm not sure. But I know in those days they used to see them as demons or disease or something. At least I still have a friend who is a little bit schizophrenic. He got to know early. I'm very sure if it was maybe in Prof's age, by my age they're getting better. But I'm pretty sure if it was in Prof's time, probably say one witch or something is happening to you. Or there are things we easily neglect because we think they do not occur to us. So within our lab, most of what we focus on is autism per se in the midst of all the spectrums of brain developmental disorder that impact how you relate to your environment. So what are key features of these social deficits, interaction deficits, including autism, are they tend to be socially isolated, they want to stay on their own. They have anxiety, which is a major thing you see across most mental health spectrum. Depression, I didn't put that there. They tend to do things repetitiously, what we call repetitive behavior or stereotypic behavior. They're a little bit aggressive in some instances. They're not able to communicate, and then they have cognitive deficits. Autism is the most prevalent of these neurodevelopmental disorders, at least at most recent epidemiological studies. And it's a broad range of conditions. Impaired social interaction is a key hallmark, cognition, communication deficits, repetitive behaviors, among others. And many ASD patients tend to have this lifelong disability that they just have to live with and grow with. And probably, because we don't have the cure yet, over time we can get trained, particularly in the developed world when this can be detected quite early. They can grow to become useful to their society. They say they are very good thinkers, maybe because the way they see the world in a different light compared to we typical human beings see the world. The theology is not yet fully understood. However, we suspect, or at least it's established, that it involves gene and the environmental interactions or associations. I put this here, proper same decision, sorry. I just put it back. I talk about this because we think in the context of Nigeria, sometimes, a few times ago, there was one project we wrote, and when we were trying to defend a little bit, we were like, oh, autism, this is not a problem in Nigeria. And I was thinking in the context of the data that is available. Globally, we have 1 in 100 children. Globally, that according to WHO, at least as of the last report, this report was updated 2 years ago. They do the report every 2 years, so it's still 1 in 100. In Africa, these are some of the prevalent rates. In Nigeria, blocking there, a report that was done quite a while back, it was published in 2014 by Languages et al. Noted 23 per 1,000, which is about 1 in 43, which is actually quite high. And then the global brain disorder study by the Lancet group, and Nigeria was seen as the third country with the highest most autism cases globally, behind only China and India. I can't remember the one that comes first in between the two of them. So, then, but to us, we still see that it's not really a problem, maybe because we've not really looked at it and tried to understand how these happens to us or whether they happen in our environment. And I think I'm probably going to say also that in this context too, it is negligible because we still don't have stronger ways to monitor it. For example, this study was 2018, it was published, but it was based on a study of cases that were reported from 2010 up to 2016. Unlike in the US where they do this every year. Every year they report the previous 8 or 10 years when the children are 8 years old. So they currently, the only time they missed the data was in 2023, I think it's because of something that came back from COVID. There's already a new data act about their autism prevalence. So they have a more stringent monitoring plan, which we lack here. So I put some videos. Within the autistic spectrum, some of the impairments we see are in 3 social domains: reciprocal social interaction, lack of ability to communicate, whether it's verbal or non-verbal, then restricted repetitive behaviors. Mostly you see lack of social interest. As you see this child, he's trying to, there are people in the room, people he's both familiar with and people not familiar with, because of course, he's not ready to even listen to them in the first instance. We also have this one, we tend to see sometimes they have these repetitive behaviors or just like you see, and sometimes these repetitive behaviors can sometimes exhibit self-injurious behaviors. That's something we see with autism spectrum disorder. So, why are we trying to talk about autism spectrum disorder, which is basically a human problem, and we're trying to understand it in once? It comes from basic science or basic biology. The basic scientist, which I am, even though I'm trying to gain some skills in translation, the course I want to take towards the end of the year, foundation in clinical science, because you still also have to give yourself to be able to marry both. But now today, I'm going to talk as a basic scientist. If I train to the translation, fine, but everything. So we see that in trying to understand humans, sometimes you come from the simple to know the complex. And social interactions have always happened even in the simplest form of living things, beginning with bacteria. In bacteria, you have this thing called quorum sensing, whereby bacteria are able to, their cells basically communicate themselves and understand that we are too much in this place. It's something that helps them mitigate cell density, too much of them, so that they can expand and live in favorable environment. That's quorum sensing. You come to new vertebrates like you have the C. elegans and the Drosophila. They have this way they interact and they come together or they separate depending on whether there's food or not. We also have social insects like ants. I'm sure you've seen ants in your house. Sometimes people say there's this paper that one read, somebody wrote something about the kiss of the ant and the juice they exchange from their, I don't know what to call it, mouth, but it's not mouth, whatever it is. And they're able to tell them, okay, they talk to themselves. Have you ever found it very funny, just in 2 times? You don't know what, boom, boom, and everybody goes the same way. Before you see, everybody's back in one place. So they have that interactive skills. And then they also have, same thing, most of us work with rodents, and I'm sure you've heard about a lot of grooming behaviors within rodents, whereby they take care of themselves or clean their fellow rats when they're dirty. Those are all social cues. And of course, with us, humans, and our complex, heavily complex social interactions. So we've seen that there's an evolution of social interactions across the animal world. And actually, social behaviors have evolved across the species. But what drives these behaviors at the molecular level is basically something, for me, as a basic scientist, I'm particularly interested in to be able to infer on how those changes may impact brain development that have to do with these social interactions. Social behavior is not just an evolutionary trait, it's wired into our synapses. I think you can see it all the way down the evolutionary tree. However, our disrupted synapses, the missing link to understanding perturbations that have to, or rather to understanding deficit disorder that has to do with social interaction deficits. In fact, perturbations at the synapses are crucial hallmarks of autism. A little bit of biology, or I don't know, neuroanatomy now, I don't know what to call it, neurophysiology, whichever suits you. So, we all know neurons, and we all know that neurons meet here at the what? At the synapse. When the neurons, or rather when neurogenesis starts and then synaptogenesis, which is formation of neurons, begin, sometime about 36 weeks post gestation, you start having synapses come together. So mostly, many synapses are formed. As at 6 months to 2 years after, many synapses are formed. But beyond that, the brain doesn't need all the synapses. So it has to prune, have to start killing off, remove some, and then to fine-tune. So that's what happens. And for these synapses to be formed, there are junctions. So junctions have to meet, right? And like even when they meet, something has to hold them together. We call those things synaptic adhesion molecules, or cell adhesion molecules. And there are quite a whole lot of them, but 2 are very important. That's what I'm trying to show you. So you have this synaptic connection where we're trying to go to know. First of all, you have target synapse. Synapse, okay, there is based on several molecular targets. You want to form a synapse here, the neurons will come together, and then they will get closer, and the synapse adhesion molecule will pull them. That's when the neurons, the synapse mature, and they are held together for as long as we need. So they are called synaptic adhesion molecules. And there are a lot of them located at the synapse, whereby they are able to hold the synapse together. But our focus in our lab, and maybe because of their importance, is on these two, neurexins and neurogenes. These two, I try to call them like this. Sorry, I will have to treat, don't mind me. And I'm a basic scientist, but I'm also a good teacher. They are most like this. Basically, that's what they are. One are the presynaptic, if you still remember your biology, that means the neurons before the synapse, and one are the postsynaptic. And they extend their processes into the synaptic junction, and they are held, they basically hold the synapse together. Funny, some other things will hold this one this way, some other things will hold this one this way in the cell. So it's a whole lot of chemicals together, so a whole lot of proteins together. So we call the neurexins and the neurogenes. Neurexins is the presynaptic one, neurogenes is the postsynaptic, and here it's just a pictorial representation. So what do we do in our lab? Prof was saying something about brain neural research. When I first started out, although I've really not changed, I'm still doing neurotoxicity. I'm just doing neurotoxicity in different lights. I know the first time I worked master brain science, I know he had forgotten. That was the first time I met. I actually came to his lab to get colaveron. We worked on his colaveron on induced methamphetamine. I came with Professor Polycarp Unha. We came for some, he just picked me along to go for one PhD defense. Professor Woye, now Professor Woye, when he was in his PhD defense. We just branched the lab, and I had to come back myself later. Dr. Dedara helped me extract it at that time. So, but our focus then was on methamphetamine toxicity, but we just wanted to see what colaveron could do to it. Over time, migrated a little bit, but I still do more of neurotoxicity. More of what I do now is environmental neurotoxicity, because my questions are, how the environment, high environment, the high focus of meta, interfere or interplay or interact with the genes to affect how neurons either grow or rather develop or how we behave funny in a social context. So I studied this gene-environment interaction between changes in genes that are responsible for autism and then the neurodevelopmental perturbations we see with metals exposure. So we already know that loss of functions in these two stuff I've talked about, neurexin and neurogene, as one of the critical hallmarks we see in autism. So our current project, which is basically, I was saying that my data is a little bit minted. Minted means, even as at yesterday, my postgraduate students were sending me the last set of data. So permit me if it all looks so or a little bit bamboozled a bit. So they are about to defend, so I need to pursue them and get new ones. So our project basically hypothesized that some of the behavioral and structural deficits we probably see following mutations in areas in this gene may, simple hypothesis, may be worsened when there's an environmental toxicant. In this case, our focus is on metals, so we've not tried other things. So for these, we are leveraging the ones. So why ones? You can remember Ruskin and the 2024 guys. What most of you don't know is that their Nobel Prize was won based on the work they did in C. elegans, in worms. What everybody is shouting today, mRNA vaccine, they started from C. elegans. At the time they even did the work, people dismissed the work. In 1992 or 1993, that was when he first published the first work, something around the LET9 gene, that was what the gene was called in C. elegans. And people were like, this gene is not, we can't find it in humans. It's over after about 10 years, they repeated it until they seen homologs in humans, and that's where they came up with mRNA. Instead of before we now had mRNA vaccine. The people who made the vaccine actually got Nobel Prize before that. I think we got the mRNA, it was 2 years ago we had Nobel Prize for people who made the mRNA vaccine. It was last year they had Nobel Prize for even discovering mRNA, which is the basis for that vaccine. So why not? They're transparent, They are easy to grow, short lifespan, simple nervous system, just 302 neurons and 56 glia. They don't have too much trouble in that compared to your almost, who can remember how many we have in humans? Almost 1 billion neurons and glia is up there that we still don't know what most of them even do. I think they've been on the human brain mapping for over 15 years. It has not finished the maps. The one month had the maps completely. It was finished 2019 or something like that. So all the synapses, all the neurons, even if we don't fully know their function, we know where they are and where the synapses. So that, again, we have over 80% knowledge. So, and that approach is very simple. This is actually a video. I'm not playing the video, I just took the screen down. One of my students did it in the lab. And I called the video The Kiss of the Worm. We still don't yet know what, but that's what we choose to call it. It's somewhere on our page. It's a video that features on our website page. And what we basically do is we take CIDANs with mutations in the genes of interest that are interested in. In this case, 2 genes, neurexins and what? And neurolecins. These are genes that control what? Synaptic work, adhesion, hold them together. I made these box around it because most of the data I'm going to be sharing are based on neurexin. The neurolecin guy in the lab is still working. So, and what we do is simple. We take these worms, we age synchronize them, just like when you work with rats. You work with the same age, right? You say they are 2 weeks old, is it 2 weeks if you are using small ones? You know what we're trying to say, 3 months or 3 weeks old are the case. So we age synchronize the worms by what we call bleaching. We actually force all the worms to lay their eggs at the same time so that we know that we are starting with age synchronized. And then we simply just expose them to metals of different. Currently in the lab, we've worked with manganese, nickel, and currently doing some works on iron. So we watch and then see a few things. So that's basically our approach. A few things to know is between 15%. When we knock off, or rather when we have ones that have enough of for this NRS1, NRS1 in the one is the same thing, is the homolog for neurexin 1 in the human. So that evolutionary trend is there. When we knock them off, we realize that somehow they have a delay in becoming lava 1. Eggs become lava 1, 2, 3, 4 before they become adults. Lava 1 is they start crawling, at least for the cell. So they have this delay. We actually expect that for normal ones, above 50% should have hatched by about 9 to 12 hours. Above 50% using the bleaching method. It's not the natural method, but above 50%. But here, we see that there's this delayed response when you have either NRX1 or both are NRX1 knocks off. They're just knocked off in different alleles. That's different variants of the same gene. That's what we do. So there are 2 different things. So another thing we tried to also observe very recently is we also noticed something. So 1, they get delayed to hatch, and 2, they get a little bit delayed to reach full adult. You know, not like they still get to adult in 2 years, but they are not as long. The average height of the worm is about 1 millimeter. But these are not as long as we expect them compared to the wild type one. So they get a little bit shorter, not diaper, not dwarf, but they're a little bit shorter. And that's what we also notice. So when we measure their length, basically what my students usually do, get the ones, treat them with metal. After 48 hours, we take a whole lot of pictures, we do a whole lot of computational stuff, and load the pictures into image and do some length measuring to try to get that. This is a whole lot of. For one study, you probably do about 15 to 20 ones, and you have to do that study for like 4 trials minimum for it to make statistical sense. So we see that post at young adult stage, NRS1 reduces the body length or the developmental rate of this one. So, although another thing we notice is that we do expect with metals for that to happen, but we notice that when you add metals into the meat, these effects are worsening. Basically, metal equation worsens this effect. This study was part of my postdoc, funny, and I did my postdoc. I did 2 projects in that 1 year and a month. I went for this, and the Mickey's lab, astronaut lab, they always has these worms. I went for a rat study, because Ibro funded the rat study, I must do the rat study. But you know, we Africans now, we can walk. I remember I get to the lab sometimes 6 a.m. I leave by 12 midnight, and I'm back by 6 a.m. the following morning, so that the janitor knew me. Even till now that I go as visiting, we always greet each other, because anytime he comes to clean, he's very likely to meet Marvels in the lab. So during that time, I did 2 projects, the rat work, but the CNPS work was what really made an impact into what I'm doing right now. And then we saw that when we treated cellulars with metal, they were reducing in their ability to move, their locomotion rate, 48 hours post-treatment. We also see that here, when we do that currently in our lab, when we treat them with metal, surprisingly, like I told you, it's limited data. So some of the interpretations are still a little bit phased to us, or we'll try. Surprisingly, when NRX worms get, when we knock off the NRX in the worms, we do not see that reduction in locomotion. Rather, we see an increase in locomotion. We just like it in lab for now. Like an hyper locomotion, they move more. We are thinking it could probably do something to do with the repetitive behaviors we tend to see with disorders to these 2 genes, the neurexin and the neurogene genes. The other thing we also found out, this is currently a work that is under review in VXC Neuroscience. In this case, worms naturally don't like ethanol. Ethanol is the home poison. Sorry, I need to move to like small, but ethanol is poison to them naturally. So they would move away from an ethanol environment. However, we noticed that with these worms, where we knock off NRX, they are graduating towards the ethanol environment naturally. And it's more like that's suggesting that they have a little bit of deficit on how they sense their environment, because they sense by chemical sensation. They are not human. Even you also sense, when you smell, it's a chemical sensation it's now we know that it's chemical that you smell, odorants, and then they tap into the olfactory receptors in the brain and go on. So they are chemical sensations. So another thing we get into that. So how do we teach worms? There are worms, how do we teach them? We know they don't like ethanol, so there's something we normally do. They live in soil, rotten fruits, that's their natural habitat, and they live in a kind of saline environment a little bit, and they tend to gravitate, if I remember correctly, they tend to gravitate away from, gravitate towards soft environments. So what do? We starve them in a salty environment. In this case, the salt is just sodium chloride. We starve the worms. We want to teach them that when you're in a salty environment, you will not see food. I don't know if you get it. They eat bacteria, so we remove bacteria, put salty environment, we keep them there for like 4 hours. The normal one will learn, but when I take them back to an acid phase whereby you have a salt environment and you don't have a salt environment, they will gravitate towards the non-salt environment because they associated salt to hunger. And that's what we would see when we have ones that are not either treated with metals or that are not mutated, as we see here. But another thing we noticed with our, though they are not yet significant, we still have a few trials to do, is that these ones that are either knocked off with NRS or that have been exposed to metal fail that test. They are not able to clearly detect that salt is associated to hunger and not in the context of that experimental setup. Okay, so that then we did something. So worms also do something. I'll bring it down to it. They said mothers are very sensitive to their young ones, right? If there's no food, let's say there's only one bread, and the mama gets, pardon me, I'm from Wari, so sometimes the pidgin English just flow, and I like it. And the mama gets like 2 kids, and there's only 1 loaf of bread. Mothers don't eat first. What do? They will give the food to, it's normal now. They don't some Facebook images. You always have, the mama will collect small. So what do we do with the worms? It's a natural tendency for adult worms, they are not male or female, they are hermaphrodites and males. But it's a natural tendency for adult worms to move away from the bacterial worm when their progeny starts multiplying. They will come back, but they will move away first and then come back. We call that food living, because they understand that, yes, the young ones, when I use the word understand, they're making it look big. They don't really understand, but you get what they sense, please, that young ones have to eat. However, what we noticed with these ones, day one, we do day one, we don't have as much progeny, and then the two adult ones. We noticed that these ones that have our NRS gene do not really live as much as the control, or as much as the wild type. So, in our heads, we are thinking that we have this deficit in social dynamics between the offspring and the what? And the parents. I'm almost rounding up because it's supposed to be a very short lecture because I also want feedback as well. So, this is something we do. Which one is this? Okay. Want to do something. The borders, we call it body. This is actually a one that actually shows exaggerated body. Something about the yellow peptide regulator that what is homologous is when we delete it, the ones tend to stay on the border of the bacterial lawn or the bacterial plate. So we actually basically use it for control. And we noticed that when we exposed, not exposed in this case, our worms carrying our NRH loss of function fail to, or rather they have this tendency to become towards the border, which we see here, compared to control ones that are basically everywhere. So we say that they show an increase in bordering compared to white type. And we are inferring that this lower bordering symptom may be one, either a lack of social interest in going around in going to other ones, or two, could be a lack of trying to explore the environment. They are comfortable with where they are. A few things we've done, those are most of the behavior. A few things we've done, in the lab, what we try to do, we labeled ones with mitotracker. Mitotracker is this case, mitotracker red, is a dye that helps us monitor mitochondrial health or mitochondrial dysfunction. And our focus is because, apart from the known things of oxidative stress and inflammation that typically happens to almost all brain diseases, mitochondrial dysfunction is a major marker for autism. And we noticed that when we give metals, so we didn't see any difference between whether it's a meta or NRS loss of function. We just see an increase in mitotracker ALA or mitotracker dye, which basically points to increase in mitochondrial derived forms or in impairment to how the mitochondrial membrane is able to separate or filter things within the extracellular space. So, however, in some instances, we see that this combined effect does exaggerate this impact that we see with mitochondrial derived forms. And this is also something from that previous paper, something we are trying. So we take ones. We have these ones that give out light, GFP. Some of you should know what GFP is, green fluorescent. They already give out GFP in several neuronal populations, in dopamine neurons, GABA neurons, glutamate, cholinergic. So we take them, we cross them with our ones that have our mutation to generate a new strain of one that is carrying a GFP in a particular neuron and also carrying the mutation of interest. We've only been successfully able to do that for dopamine. We're still trying to do it for GABA and cholinergic neuron because our interest is sometimes, most of the perturbations around the neurons actually impact how ASD patients behave. For example, GABA neurons modulate inhibitory and transitory synapses that impact that impact some of the clinical symptoms we see in ASD, or dopamine neurons also impact the stereotypic behaviors we tend to see with this ASD patients. So here we see, we see that across all board, we show an increase in dopamine generation following metal treatment in both NRS1 and NRG. I took most of the data of NRS1. This is already a paper, so we have a little bit of, or rather a paper in review that has energy loss. So I've been talking a little bit of science. don't know how much time I have left. I should be rounding up, so it's going to be a short one. What are my summaries from this? The molecular data indicates that autism is becoming a national burden in Nigeria from some of the prevalent epidemics, still largely neglected. Maybe neglected because one, we don't do continuous data, two, we still don't have our stocking, which is what Professor Science says. Sometimes we also want to do translation. And there's this, I've always worked in National Lab, but now I have a visiting assistant. I've been there for like 2-3 years, so I am trying to spy into, if you were into other people's lab. And there's this group that work with humans and ASD, and at least we're trying to, the project we're just thinking, for now we're in the dreaming phase of trying to understand how we can keep having monitoring data for autism in the country, because some of the things we identify is we don't yet have the capacity of the manpower to really diagnose some of these, so maybe that's why it's largely neglected, but it's becoming a national body. If I try to think about it like this, if 2016 was like 9 years ago, we have a 1 in 43, we don't know yet what we have. Hopefully it's just as it is. Or maybe the 1 in 43 will not get diagnosed. We don't have a full complete picture. Synaptic dysfunction, particularly from disturbances to this cell adhesion molecule, are critical hallmarks and majorly trigger the resulting social deficit and structural impairment we see in ASD, particularly that have to do with the neurexin and the neuroleptin gene. So we have saying that C. elegans model could offer one, a rapid and relatively easy model for us to understand how molecular perturbations occur at that level, and possibly, if we are obtaining proper parts, screen potential therapeutic compounds. In this context, okay, let's show you some of our future talk. Some things we are developing and some things in the pipeline waiting for 1 or 2 grant funding. We're trying to investigate the kind of it's still part of this program, in multi-neurotransmitter systems in ones. We've done dopaminergic, we want to do GABA, and we want to do choline. We don't without metas. We're also trying to analyze whole transcriptomics using RNA sequencing to better unravel maybe any new molecular pathways that may be impacted when you knock off these genes. And this is a new one, something we have somewhere in the pipeline. We want to understand how maternal and or paternal, though mostly maternal, but the paternal is an interest for sometimes in the future. Maternal and or paternal psychosocial conditions, the schizophrenias, adult schizophrenia, and all the rest, how do they impact offspring's synaptic health that may be relevant for autism? And finally, we're trying to also look into the future to identify compounds that can target mostly from indigenous local plants that we can use to target some of these chemical perturbations at the level of the synapse that may be or serve as an effective strategies for autism. There are a whole lot of acknowledgement to go through, but I would first of all, everybody in the lab. I said I wasn't going to have more than 3 students when I was starting off, but right as the lab is full. The one thing bold that basically Dr. OK in Germany happens to be the other German. My wife, she does a whole lot of neuroscience, and it's basically how what we work on together that is currently on review from the lab. And then these 2 students, Victor and Tohib, actually drive the master students that are driving most of the experiments you see here. And our collaborators across, and then some of our funders, which we have quite a lot, and we're still looking for more. And with that, I say thank you.
Thank you very much. Please, another round of applause for I like the way my brother teaches. He doesn't give lectures, and that's something that I also try to, I'm sure that everybody has been able to pick a lot of things from what he has said. Because what he talked about is very esoteric. It's totally not the usual. But he has been able to deliver in such a way that it makes it sound as if all of us understand what nematodes and worms are. I don't actually, but so observe a couple of things. I work on gene-nutrient interactions, as you know. So one of the major things that we have now is that most of the factors that affect us, most of them are not genetic, as you know. which is why it's very important that our research now should start trying to drive in that direction. This lends more credence to why work like this are very important. Many of the things we are eating, the things we are breathing in, the things that are actually the ones that are causing most of the diseases we're having now. The time has passed when it was, things that were transmitted and passed from your parents down to you. Most of them are now things that are around you. And I just learned today that apart from, I always thought that diet was the most important, but now they are just making me understand that mental exposure can be a major factor that can also affect it. A couple of things that I also want to, just a couple of summaries. First, I wanted EST in, because this is something that I think about sometimes. I'm not a neuroscientist, and I keep asking you guys very strong questions, as you know. If 1 in 100 is a prevalence of EST, for instance, is that not lend credence to my sometimes interpretation that might be normal also? So these are things that you should start thinking about. Like, this is another categorization we just haven't figured into our categories yet. So, because autistic patients can actually be, or autistic children especially, if you are treated right, can actually be some of the most brilliant people in the world. So, is it not a categorization that we have missed out because we have taken it long as, oh, this is how normal people should behave, and then these ones are behaving in a different way. So, I think these are some of the things that you might help me to understand a little bit better. I also think that...
I'm not like Prof.
Yes.
I'm not mature to left way. Prof, so, Prof has been doing this for how many years now? So, permit us that are still coming up little by little. So somebody will say, she is also still coming up. So what will I come up? I'm still coming up. So, true, we now have something we call neurotypical individual. So, I think, I can't remember where I was reading it. There is actually something called high functioning autism. Meaning, if you know the amount of people I'm suspecting for high functioning autism, thank God it's not with that right. Meaning, they are autistic traits, but it's not serious enough to have a debilitating effect. And those are the kind of people most of the time that have this high IQ, because somehow they're able to view the world in different abstracts that you, neurotypical, the normal human beings, do not see. So I do agree in that context that we also have to, like say, 1 in 100. It's quite a whole lot. In the US, the current data is 1 in, I checked it last week when we were writing 1 gram. 1 in 63 or 1 in 53, that's the US. So they're even getting close to our old 1 in 43 data per se. But like I said, we still don't know what that current rate is. So,
thank you very much again. So we will still take questions and answers, but please let me again ask him another question, because this is my chance to catch him before he leaves for med again. Now, I was, when you were talking about long distance, one of the factors that I know very, very well has an important function in social conditioning is cortisol. I mean, glucocorticoids basically. They practically affect how you bond, how you interact with people. And I saw some of the things you were, describing that. Is it something that you can, consider in the future? But you need to try to look at how, or do worms exhibit any kind of glucocorticoid, activity? Yes.
Okay. I'll just say something, but I won't say more because I don't know.
So, I do something in my lab with my students. And my prof, I keep referring to prof, I felt like that he had his lecture before me because I could mirror a little bit of things. There's something called training. You get trained well, you train other people better. You cannot train like the thief in Vicky's lab, come on, it's way. And he had this way about the way he trained us in the lab. There's a big project. This is the big project that the funding is coming from. You come, you look at the project, you find the part of the project you can cut from it, because that's the justification of keep receiving that funding. You do your own. You find so. With my students, I try to do that. Recently, these 2 students that are actually powering this work, on their own, they just woke up. I think we wrote a paper, a book chapter or something about stress, evolutionary trends around stress with the C. elegans. And from that, like I said, I don't really have data, so I can't say anything. But we are trying to use the C. elegans to mirror stress. That practically, I think they are doing it by
doing an heat shock stress, and the thermal stress, thematic stress. You see, I'm still strange because we don't have time to discuss it. They should finish the MSFS, let me pursue them, and then we can come back to it. But it's also another way we can look at it, because yes, in all factor, most of the social control, social behavior, or how social interaction is affected is basically based on stress. In fact, I will just give a very simple, I like it very simple. Okay, we understand. The married people, we understand. You are tired, you are stressed, whether you are a man or a woman, husband or a wife. The day you are stressed, you come home. You that have been greeting good morning and you've been smiling to say good morning, but the day Dr. Deji offended me in the lab and I carry myself home, they greet me good evening and I react differently. What is that? Stress impacting your what? Your social interaction. So, now, let's go.
Please, if you have questions, you can either prepare to ask your question or write them down as we take the last phase before the question and answer.
So please, like I said, keep your questions. I'm still going to ask one last one. This is my opportunity, like I said. So there's something I was looking at. You had this micro tracker. It's something I'm quite familiar with. So I was looking at what you're doing with it. Fantastic. I mean, I'm checking what happens with the mitochondria and how its activities. But then, one of the key functions of the mitochondria is actually in metabolism. It's something that helps you to metabolism. It's one of the major functions of the mitochondria. That's the key function. So one of the things that happens is that it's able to change its own DNA. We have mitochondrial DNA. It's able to start switching around its own DNA in such a way that, depending on how your metabolic requirements are. So one of the things that I just struck my mind was, what it might just be, if what you're saying is in that direction. Some of this social dysfunction or disorders might just be totally related to how the body is able to generate energy. Which then, how the body is able to generate energy. You understand this? For instance, if you do not have enough energy and the brain does not, let's just say for instance, you haven't eaten for 2 days, how easy it is to study? Now you understand. So you get my point. So that's another direction that I was thinking about ICAP. Does it make sense for you to start looking in terms of our head? Anyway, while you're thinking about it, we'll take the questions.
Okay, please. Questions? Please, questions. Anyone with questions for the presenter? Please roll. Don't let us miss you, please. Questions? Are you reading off your own? Okay. Another question here. 2 from the other end. Any questions, please? Okay, so we have 2. Please, can you come forward? Oh, sure. Your name?
I'm I have a very simple question. When you were presenting, you talked about putting worms in a salty condition and to trigger hunger, is it hunger or lack of food? So I want to ask that because normal worms are told when we put salt, they will die. So
I mean, I was mixing it up. So, worms don't like salt. So, we condition them to hunger. Condition them to hunger and food in salt. Even though they don't like salt. But because they've been trained that where the salt is where the food is. They will gravitate towards the salt. The salt is not the one that will kill them. It's just small, something, it's 5 millimolar sodium chloride or something like that. It's not like it's really salt. We just have to teach them salty environment, no salty environment, with food, without food. They learn that, okay, for today, even though we don't like this environment, it's good enough because there's food.
Thank you, sir. Good afternoon, everyone. So this is my question. what are the early signs of autism in children? And also, what are the effective strategies that we can use to support individuals with autism?
Disclaimer, I am not a clinician. I do know that is a good disclaimer. So, I am not going to attempt. There's a reason I want from how children behave, how they repeat, how we know all kids. Sometimes that's where the argument comes about. Maybe that's just an atypical. There's a whole lot of diagnosis scale that comes into how autism is diagnosed. Disclaimer, I don't know much about it. I'm sure that my program officer is insisting, program officer is the person who I want to describe them at now, who monitors your progress in an NIH grant. She's insisting that before my 5 years is over, I have to go and take a clinical course in autism. She's not like insisting. She's saying you mentioned it during my review, and okay, we talked about it 3 weeks ago. I was like, okay, I'll find a way. So disclaimer, I really don't know. Strategies for how you are able to mitigate them. I can only speak to a lady in the context of early diagnosis. Sometimes that is, to a large extent, that is what we miss in this world. I have a friend, my colleague in Wiki back in the US, Professor Asaf, he has a child that has autism. Within 2 years, at least that's the last time I spoke to him, the child is better. Most of the time for us here, there's no diagnosis. You just grow over and get what, because nobody is even thinking about it in that context.
Thank you, sir.
Okay, going to the last part of the session, which is the presentation of the award. So I'll call to the podium Professor Barondi to help us present the award. Thank you.
Well, I remember the story now when it came to Ibadan to collect a collaboration with that professor. I remember the first time of meeting him, and the last time, the second time was during a project, a national project, a defense, in Abuja, where I stood as one of the assessors, so those to give the grant, and he did very well. Was able to present. There is something too on this autism thing, and I think I won't ask those questions. I'm asking those questions that day, which he has taken and working on. I'm very sure we also enjoyed the presentation, and we have been impacted. So the Merit Award of Recognition is presented to Dr. O.A. Ijomone, a plenary speaker at the first SBMS conference at FUTA, in recognition of the illuminating plenary session and impactful research contributions to neurobiology and toxicology. Dr. Ijomone has worked well with Professor Ashna in Albert Einstein University. Ashna is also my very good friend and my collaborator, and as I was telling him, we used to speak at an international conference in Jackson State University, for so many years until the grant is exhausted, and we stopped meeting, from 2018. So hopefully we'll get some other grants so that we'll be able to meet again. So many congratulations to you for the excellent work.
John, I'll put at the back from Professor
All right, so before we close the session, let me just say something about the spectral disorder for the person who asked. So we do not have, it always changes what we're using in diagnosis. So you need to be careful. There are lots of people that have been diagnosed with autism that are not really true. So, and there are different, that's what we call it the spectrum of disorders. There are too many things that feed into it. So you need to be very careful. By not just putting the doctor and you want to be sure that it's the right. Alright, so with this, I will declare the plenary session closed. Thank you.